Hello everyone, my name is Michael Edu, and today I'm here with my fellow ALU scholars, Bernice Ajewa, Carolina Perez, and Mercy Datsuban. Our mentor is Dr. Regine Tian, and our project is detecting the ALU polymorphism in human DNA. So now let's talk about ALU. Now, in our DNA sequence, only 5% actually codes for proteins. The remaining 95% are called the junk DNA, and we think it doesn't do anything. ALU makes up about 5% of this junk DNA, and the history of ALU is that it is actually a retrovirus, so it's related to the AIDS virus, and it affected our ancestors millions of years ago, and it has been passed on from generation to generation. Now, what this virus did was it copied DNA sequences and randomly placed them in our DNA sequence everywhere. Now, an example of ALU is the tissue plasminogen activator, TPA25. Now, what it does is it codes for proteins that prevent our blood from clotting. But because it's a junk DNA, it is not activated in us, which is very good. Now, in order for us to study ALU, we have to make it visible. And so we use a technique called the PCR, which stands for the polymerase chain reaction, that will actually amplify this DNA sequence and make it visible in a test tube for us to actually work on them and to study them. Now, the PCR is a very popular technique. It actually has been used to resurrect the DNA of fossils like the Neanderthals, and that has been studied all over the place.
PCR test will tell us if there were any ALU TPA25 present. Since ALU TPA25 is dimorphic, we can get two possible results, a 100 base pair or a 400 base pair, sometimes even both. A 100 base pair will be considered as a negative homozygous ALU. A 400 base pair will be considered as a positive homozygous ALU, while a 100 and a 400 showing together will be a heterozygous. To find out if we have any ALU insertion, we have to extract our DNA by obtaining hair follicles cells or cheek cells. We conducted our experiment using cheek cells DNA. Rinse our mouse with saline solution, since salt keeps the cells in proper osmotic balance so it won't burst. Then the cells are spun in a centrifuge and boiled with Telex. This causes cells to disrupt and the DNA to be extracted in water.
When the DNA and the master mix and the positive controls are being put into the thermal cyclase to amplify the DNA target sequence. PCR amplifies our DNA, and this slide wants us to know what is needed for PCR. TAG is a nickname for a bacteria that survives and reproduces in hot spring. TAG polymerase can survive in extreme hot temperatures, and this enzyme does not denature like most other enzymes. Inside the PCR tube, we have the template, which is the DNA that we want to amplify, and we have the primers, and we have the nucleotides, which will match with the primers, and we have the magnesium ions, which without it, it will help all these reactions to take place, and we have the buffer containing salts, and we also have the tag polymer. Once we have the primers corresponding with the particular nucleotide, we put them into chains. That is where the tag polymerase come in. PCR work by heating to denature the DNA strands, which is to separate them. Separating the strands will enable the primers to set in. We code to match the primers to the template. On our studies, it will be on chromosome 8 between axon 8 and 9. After primers match with the templates, we warm it to activate the tag polymerase, which extends the primers and replicates the DNA. Repeat the cycle about 25 times to get the required results or sequence that we need. One cycle usually takes about 2 minutes, so 25 cycles will be like 15 minutes. After amplification of DNA, the DNA is put into a gel to see the ALU insertion, whether it was inserted or not. We had 10 samples. 5 were heterozygous ALU insertion, which is a positive and negative. 5 were homozygous inactive, which is negative. Base pair is always 100 and ALU insertion is 300. So if it was inserted, it will be 400 base pair. the 5 heterozygous allele insertion, you will see that their basal pair has risen to 400 BP. The other 5 samples which didn't get any allele insertion on chromosome 8 remain on the 100 BP DNA marker. To conclude, we have successfully identified our allele genotype with PCR and gel electrophoresis.
Allogenotypes can be used as identity markers for forensic and paternity analysis, also for studying human ancestry, evolution, and genetic diseases.